So thank you everyone for joining us at this tutorial. And I think that many of you probably joined us at the Open for Neuroscience Symposium and heard talks about the Allen Brain Observatory, and you're feeling inspired to use this data set yourself. If you missed today's symposium, you can find the link to that on YouTube. We'll share that, and it's going to be available if you just go to our YouTube channel at youtube.com/Allen Institute. So here is what we are going to do today. So this is our introduction, and hopefully your AWS instances are spinning up for those of you who'd like to follow along. Again, if you are watching this tutorial afterwards or would just like to refer back to it, you can run all of this code locally, and it's available on our GitHub. Saskia is going to start by going through how to work with our 2-photon data set, and then Josh will work with our Neuropixels data set. And then we have time for Q&A as well. So please be posting those questions in the Q&A window here in Zoom or in the YouTube chat. So again, please place all those questions there. Please use the chat to talk to each other about your research interests and have other comments, but please don't put any questions there or else they might get lost. And make sure you're sending those chat messages to all panelists and attendees so everyone can see them. And we have live captions here in Zoom, and they'll be available on YouTube as well. Our presenters today are Saskia and Josh. Saskia is an associate investigator and Josh an assistant investigator, both in the Allen Institute Mindscope program. And they were involved in the development of what you're going to see demonstrated today. So in case you missed the symposium this morning, the Allen Institute is a biological sciences nonprofit located in Seattle with focused research areas not only in neuroscience, but also in cell biology and immunology. We also support cutting edge research across the world and across the world of biology with the Paul J. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental sciences. And our approach is guided by 3 core principles, which I'll return to in a moment. Our work generates data, knowledge, and analysis tools, all of which we share publicly, including the ones that we are sharing today. And to date, we've collected about 14 petabytes of data, which is 2.8 million DVDs worth. So the most relevant of the core principles today is, of course, open science, which is that we share all of our data and the analysis tools that we're about to talk about publicly for you to use in your work. So please check out some of the other webinars in this series, as well as today's symposium talks about research using these resources to learn more. Again, Q&A for questions, chat to chat with each other. We will be monitoring YouTube for questions as well. And we'll post a link afterwards in the video description for you to go to our community forum and ask more questions after the fact. So Saskia, take it away.
All right, let's get this back up here. Again, you can see my notebook. Great. All right, so I'm going to start working through this. There's several sections here, and I really only want to get through the first 2 sections, which introduce you to kind of how to use the SDK to access the data. And then there's some sections below that on computing tuning curves and looking at correlations that I'm not going to try and get through today. Just I want to make it so that you can access all of the data that you're going to want to that you can get out of the NWB files. So to get set up, first we're going to import some standard packages, NumPy, Pandas, Matplotlib, and then we're going to set up the Brain Observatory. The key thing here is that there's a manifest file. If you're working on your local machine for the first time, you would just get rid of this call for the manifest file, and it'll create a manifest file. And that manifest file then tells it where to download data to. And once data is downloaded, when you use the SDK in the future, it'll point to the files that already exist. So if you use this on a local machine, the first time, leave that blank. The second time, point it to where that manifest file is located. But here I've set it up with a manifest that's in this AWS environment. All right, so what I've instantiated now is this Brain Observatory cache that then looks at that manifest file and which has all of this information about the data set. And we can use a number of functions to kind of find out about the parameters of the data that we are able to access and that we'll need to use in order to kind of find specific experiments and specific data sets. And so there's some functions that kind of help us do that. So we want to start by, what are all of the areas that there's data been collected in, and to we call these visual areas or brain areas, we call them targeted structures. These are structures that were targeted for recording. And so we have a function called get all targeted structures. And if I run this, what it returns is a list of strings, and these strings are the names for all of the areas that we've collected data from. They all start with the prefix viz. That means visual cortex, and then they have, this is area AL, area AM. VisP is the primary visual cortex, so that's what we call V1. And so these are the 6 visual areas that we've collected data from. We have similar functions to get other information. So for instance, if we want to find the imaging depths, if we want to see what are all the options for imaging depths where we've collected data, you see there's a lot of different imaging depths that different data sets were collected from. We also used a lot of different Cree lines. So again, I think you probably can notice this pattern of get all X, get all Y. So get all Cree lines gives us a list of the Cree lines. And this is one of the things that's valuable is that, if you want to use the Cree lines, and I'll show you how we do this in a minute, to identify a particular, say you want to get data that's from the VIP interneurons, you're going to need to pass it this exact string. And so it's good to kind of have this as a reference function so that when you forget, is it iris with a lowercase r or a capital R, you can get the exact string just by using these functions. I'll make this a little bit bigger. All right, so those are all the Cree lines. We also used a couple of different reporter lines. So here we get a list of the reporter lines. So the reporter lines, that's the GCamp 6 indicator. And for most of the data, we use GCamp 6 fast. And it was either AI 93 or AI 148. These are different constructs of this calcium reporter. For a little bit of data, we used GCamp 6 slow, which was either AI 162 or AI 94. Depending on what you're doing, you might want to target the slow data, or you want to explicitly exclude the slow data and only focus on the fast data. And so you might want to use the reporter lines to select data sets based off of that. Let's look also at the different stimuli that were used. So get all stimuli gives us a list of the names of the stimuli that are used. And these stimuli, if you saw my talk at the beginning of the morning, not all stimuli are presented in each session. And so we also have this organized with different session types. And you'll get a list of the names of our different sessions types: 3 session A, 3 session B, 3 session C, and 3 session C2. And the difference between C and C2 is that partway through our data collection, we found that our locally sparse noise stimulus, which had a 4.5 degree stimulus spot size, wasn't very effective in the higher visual areas. And so we added in addition to that 4 degree spot, we added another part of the stimulus with 8 or closer to 9 degree spots, but we call it 8. And so we renamed the session C2. And so some of the data will have 3 session C, and some of the data will have 3 session C2. All right, so you can see there's a lot of functions here just to kind of familiarize you with what's the parameters of the data set. And they really is just kind of this BOC get, we can get all Cree lines. We showed you that, stimuli, targeted structures. There's a lot of other functions that are, if you just kind of do this tab complete, that you'll see here. And hopefully most of them are self-explanatory, and then I'll get into the ones that actually give us access to the data. But you can just kind of see what these different parameters are. All right, so I'm going to now use some of these parameters to start finding some specific data sets. So you can pick any visual area or Cree line that you want. I'm just going to, for this demonstration, I'm going to look in our primary visual cortex, VisP. I'm going to look at this CUX2 Cree line. CUX2 is a dense excitatory Cree line that's concentrated in superficial layers, so layers 2, 3, and 4. So I'm just going to define these 2 variables. And now I want to find all of the experiments, all of the data sets that were collected in primary visual cortex with CUX2 Cree line. So it's another get function under the BOC, but this is called get experiment containers. And I'm going to specify that the targeted structures is the visual area that I already defined, V1, and the Cree lines is the Cree line that I already defined. All right. And so this returned a list of experiment containers, right? An experiment container, I don't know if I've defined this terminology very clearly, but an experiment container consists of 3 separate sessions. So it's the 3 sessions that we image the same group of cells. Each session is a different compartment, a different part of the stimulus. So let's just look at how many experiments we have here. There's 16 experiments for COX-2 in V1. We can take a look at, there's a bunch of information here. So I'm going to make a data frame of this list because I find that an easy way to look at it. So each row here is one experiment container. And then we've got a lot of information that's included here. So we've got an experiment ID. This is a unique identifier that we can use to access this container. We have information about the imaging depth. So you see for COX-2, we've imaged it at 2 different depths, 175 and 275. All of this is in V1. All of this is COX-2. That's how we selected this group of experiments. So that doesn't surprise us. It all has the same reporter line. The donor name is an identifier for the mouse. And so sometimes mice are used more than once, but not for the same imaging depth and Cree line. Outside it, like we collect data from the same group of cells 3 times. And then for that mouse, we might image a different visual area or a different imaging depth. But so you won't see the same ID for the same imaging depth, same Cree line. All right, and so this kind of gives you some information, again, that might be useful for subselecting data that you want from this. So I'm going to pick one experiment from this. I've already kind of pre-filled this with a container ID. This is this ID that's in this column. So I'm actually just taking the first one here. And what I want to now start accessing the data for that experiment container. And as I've mentioned several times now, there's 3 different imaging sessions. Each session has its own data file. And so we have to, if we want to work with the data, we have to identify the individual session that we want. So let's see what sessions are available. Let me scroll this for you. So now I'm going to use this function that's called get OFIS experiments. And this is going to give us a list of experiments. And I'm going to pass it is everything freezing on me. All right, my notebook is freezing. There we go. So I'm going to pass it the experiment container ID that I already defined in the line right above that. And this gives us, this is just marked down that got messed up. Let's look at the list that came back. And again, I'm going to make a data frame of these sessions. And so here we get a list of the sessions that are included in this experiment container. So again, the imaging depth is all the same. The targeted structure, the Cree line, the reporter line, those are all the same. The donor name in this case is all the same because it's from the same mouse. The differences that we see are this ID on this far end. This now is no longer the experiment container ID, but the session ID, which points us to this very specific file for the very specific session that this is. I think the other key difference that you see here is the acquisition age. So you see, actually, in this case, 3 session C was taken first, then 3 session B, and then 3 session A. So that's the order in which these data sets were collected. You can see the age of the mouse for each of those days. This little graphic just explains which session has which stimuli that you can refer to in the future. All right, so I want to get the ID for the imaging session that contains the natural scenes. And so I'm going to use again this get OFIS experiments function that we used. I'm going to pass it the experiment container ID that we already chose. And now I also want to pass it a stimulus name. So I want to identify the particular session that has the natural scenes. And so this will return, again, a list. There's only one item in that list, so I want that 0th item. Let me just make this a little bit more legible for you. I want that 0th item, and what I want from it is the ID value. And this is going to be a variable that we call session ID. So let's run this. We can now just see what that session ID is. Again, it's this 9-digit string that we see again in this list up here. It's this one here from 3 session B. All right, so this session ID is what we need to be able to actually access the data for this particular experiment. So now we can kind of dig into the part where we actually start looking at real data. So I'm going to make a data set object. And so now I'm going to use this function called get experiment data. And I'm giving it the session ID that we just identified. And this will take a little minute, especially if we're not in AWS. This might take a few minutes because that's actually where it downloads the NWB file. All right, so now we can use this dataset object to access all of the pieces of data that are in our NWB file. And so let's just start looking at what we have available to us. So we've got a max projection. And in our standard fashion, all of our functions are get this, get that. So get max projection will give you the max projection. This is an image, or it's an array, but we can look at it as an image. So I'm just plotting, I'm just making an image of this max projection. This is a max projection of the entire imaging movie, and so it's the maximum value for each pixel. And it gives you a pretty good feel for how many cells are in the field of view. It's not perfect. There are cells that won't show up very, very strongly in the max projection, but it gives you that first pass glimpse of all of the cells that are in our field of view. We also have the segmented masks that were used to identify each cell and get all the traces. So again, using our dataset object, get ROI. It's not auto-completing for me, but it's mask array is equals. And so this is an object. Let me add a cell here. The shape of this is 174 by 512 by 512. 512 by 512 is the size of our image. And you'll see, like in this max projection, right? This is 512 by 512 pixels. And so this array, this ROIs array, it's got a single plane for each ROI that's identified. And so you can see each ROI individually. Let me just look at one of them. So that is one of the cells. We could also just look at all of them. Let's try this. That's not what I want. I want IM show. So here you can see all of the cells that were identified in this field of view. And if I leave it as an exercise for the viewer to superimpose this on the max projection and kind of see what that looks like. But this gives you all of the ROIs that were used. All right, now let's start looking at some. Let's keep going, and let's look at our fluorescence traces. The data set includes a lot of different versions of the fluorescence trace. So there's the fluorescence trace, there's the neuropil correction trace, and we also have the delta F over F, where we've computed the delta F over F across the entire session. This tends to be what I defer to. So I'm going to pull this up. The function to get the delta F over F traces returns two things. It returns timestamps. And then the delta F over F array. So we can run this. So the imaging is done at 30 Hertz. The timestamps are just stamps that we have kind of retroactively applied to each of the data frames. So they're just spread out at 30 Hertz. I often just work with the frame numbers. All of our stimulus table information that I'll show you in a moment deals with which 2-photon frame was active when, or was being collected when the stimulus began or ended or what have you. So I tend to kind of ignore these timestamps, but they are there and can be useful for certain things. So just looking at the shape of our delta F over F array, there's 174 rows. This is it aligns with the number of ROIs that we saw in our mask array. And then this is the time dimension. So that's the time duration of the experiment. So let's look at some of these cells. So here I'm going to plot the first cell. So just plotting this delta F over F for the 0th item. And you see there's times when it's active and times when it's not. I'm now going to make a plot. I've pre-filled some of this just to cut down on the clicking and clacking. Here are the first 50 cells from this data set. So these aren't all of the cells, but you can see the first 50. And yeah, you can start to see that different cells are active at different times. Here's a cell, it's really nice and active, and then it kind of goes quiet for a bit and comes back. Or here's some cells that show really strong responses at different times during the experiment. So it shouldn't be a surprise why we see different cells active at different times. It really is because we're showing different stimuli at different times, and some cells respond better to some stimuli than others. So let's start looking at the stimulus information that is contained within our data set. So the first thing I want us to refer you to is something that's called the stimulus epoch table. This is really useful for stimulus epoch table. This is a useful data frame that, why this is taking a long time, that gives you a list of all the stimuli that are presented in the session and kind of the epochs when they start and when they end. So the way we did these experiments is that we would show, say 10 minutes of, here it tells us exactly, 10 minutes of static gratings, followed by 10 minutes of natural scenes, followed by 5 minutes of spontaneous activity. And then we come back and we show natural scenes again, and it's a continuation of the natural scene stimulus. And so we broke our 1 hour imaging session into blocks of around 5 to 10 minutes, where we'd show these different stimuli and then come back and keep showing more of them. And so this gives you just kind of that bird's eye view of the entire session. But we then have specific information for, and I'll show you in a moment, the stimulus table for the static ratings, or the stimulus table for the natural scenes that tells you the exact trials and when each trial began and what the parameter values of that trial were, either the grading conditions, the orientation and spatial frequency of the grading, or the image number for which image was being presented. So, but just looking at these stimulus epochs, these big broad windows of time that the different stimuli are being shown. In this cell, I'm making the same plot that I made above. I'm plotting these 50 traces of delta F over F, and now I'm just shading in the different stimulus epochs with a different color for each stimulus type. And so you can see now where we see that these cells, like this is a cell I pointed out before. Here's the cell that's really active. And then it kind of becomes quiet, and you can see that that the alignment of when it's active and when it's quiet really aligns with when we're showing these natural scenes, and then it switches to spontaneous activity and the cell stops being active, and then it comes back to more natural scenes and we see more activity. So you can start to find cells that kind of show a selectivity for different types of stimuli. And even just kind of on this course level, you can start to see some interesting things. I'll come back to this in a minute. Let's keep finding more data. So I mentioned before, in addition to the neural activity that we've recorded, we also have the kind of behavioral parameters of the animal. So in this case, I'm going to look at the running speed. So again, using this function get running speed, it returns the running speed. I'm calling this dxcm. This is the change in distance. It's in centimeters per second. And then again, it also returns some timestamps. That's the second thing there. And I can plot this running speed. So you can see in this experiment, the animal has some small bouts of running early on in the experiment, but it's mostly stationary in the middle. And then there's this window kind of towards the end of the experiment, the last third of the experiment, where the mouse runs more consistently. And we can add this running speed back to the figure that I made above, where we've got the activity of these cells, we've got the shading of the stimulus, but now I'm going to add the running speed as well to that plot. And so here now you can see we're kind of piecing all of these different pieces of information together. I think that this is a good way to kind of get a high level view of some of the things that are going on, and you can kind of dig in, and even just at this level, we can start to see some interesting things in the data. So I've picked out a couple of key cells. So that very first trace in our plot above, up here, I think is kind of an interesting one to highlight. I'm just going to replot it just by itself. And so this is the activity of that one cell. Here's our running speed. You see these epochs of stimuli. This cell is really active during the spontaneous activity and is much less active when we show the different stimuli, the static gratings, and even the natural scenes. We don't see a lot of activity. And you'll also notice there's these little gaps. They're about 30 seconds between when we finish showing one stimulus and we begin showing another stimulus. So this is probably about 30 seconds between what is the static ratings and then the natural movie that gets shown. And you'll see you see this big burst of activity there. And again, you see a big burst of activity after this epoch of natural scenes when we have this mean luminance of gray. So this is essentially the same thing as the spontaneous window. It's just shorter. And so it's not a full epoch. But so it's kind of interesting that this is a cell that's really active when there's no structured stimulus on the monitor, but as soon as we show a grating or an image or a movie, the activity gets suppressed. And this is something we see in our data set cells that do this. They're these kind of suppressed by contrast cells that I actually find really interesting. We see some kind of differences across some of the Cree lines in terms of their prevalence. But it's not unusual to see one or two of them in every data set that we have. Here's another cell that I'm going to plot for you that I pulled out from that trace above. Again, the activity of one cell, the running speed, and then the stimulus epochs. This is a cell that seems to be pretty correlated with the mouse's running speed. You see a lot of activity that kind of aligns with when the mouse is running, and when the mouse is stationary, the cell seems pretty quiet. Here's kind of a last example of a cell that's kind of the opposite, where we see more activity from this cell, and it's probably stimulus-driven activity, but it's more active when the cell is not running. But then the responses are either smaller or just less prominent when the mouse is running. So we see kind of a bit of an anti-correlation between the cell's activity and the running speed of the mouse. So in this one experiment, just in these first 50 cells that we have up here, you can already start to dig out different types of phenomenology in the data set that we're seeing. All right, a few more things that I want to point you to. In Peter's talk, when he was doing a lot of the comparison between the calcium imaging and the Neuropixels data, he talked about events. And we've done an event extraction on our data set where we take our fluorescence traces and we use an algorithm that was developed by some colleagues at UW, Sean Jewell and Daniela Witten developed an algorithm that we use to get these events. And these events have times and they have magnitudes. These aren't actually stored in the NWB file, but they are available through the SDK through the BOC. So to get these, the function isn't through the data set, it's through the BOC object. And it's get OPHYS experiment events. And again, we have to pass it our session ID. And it's going to return an array. This array has the same shape as our delta F over F array. There's 174 cells, and this is our time dimension. And so we can, again, we can plot the events. Here I'm plotting the events and the delta F over F trace for one of the cells. I've zoomed in on a particular time window. So you can kind of see what that looks like, what that event extraction did. And so we can remake that plot of the first 50 cells and the running speed using the event traces. I probably should scale this a little bit differently. It's a little harder to see, but it's the same general gist of the activity that you have there. So some analyses are a lot easier to do using the event times and a little bit cleaner to do with the event times. So that is available. But as I mentioned before, it's not in the dataset object. You have to use the BOC object to get that array, but everything else then kind of works the same way. All right, I've got a few more minutes, so I want to get to the stimulus information. We've looked at the stimulus epoch table, but like I said before, we have more specific information about the precise trials and timing of each of our stimuli. And so we're going to get the stimulus table first for the natural scenes. So using the dataset object, get stimulus table. and then we want to pass it the string for that stimulus. And this is, again, why some of those functions I showed at the beginning are really useful. So the stimulus is called natural scenes. It's not called natural scene. It's not called natural images. And you have to get that string right in order to get it. But that's one of those errors that people learn pretty quickly. And so this gives us a data frame. Let's take a look at it. Sorry. I'm just going to look at the first few rows. So each row is a trial. This column called frame that tells you the ID of the image that's being shown. And I'm going to show you in a minute how you can use that ID to actually see what the image is. But this tells you which image number was shown, and then the start and the end. And the start and the end time are in the 2p intervals, frame intervals. So we can use this start and end time directly back in this figure here, and it's in that scale of imaging frames. Let's look at the stimulus table for static gratings. Just to compare, get stimulus table, static gratings. So again, looking at the first few trials, we have a little bit of different information here. So instead of just a frame number, we've got the parameters of the gratings that are shown, the orientation, of the grating, the spatial frequency of the grating, and the phase of the grating. So we have 4 different phases that we use when we flash these images. And then again, the start and the end time. So the tables look different for the different stimuli based on what the parameters of those different stimuli are. But kind of all of this key information is here. And I mentioned that you can use this ID, this frame number, to identify which image is actually being shown for the natural scenes. And this is available in here in what's called the stimulus template. So again, using dataset get stimulus template, and we still want to tell it natural scenes. So we have templates for the natural scenes and the natural movies. These are the stimuli where we actually just showed images and movies from an array as opposed to things that were generated by the stimulus program. So, so taking a look at this array, so this is an array, it's shaped 118 by 900 by 1200-ish. So these are the 118 are the different images that we show. And so we can look at one of these images. I'm just going to pick one of the numbers. Let's take this first one here, the number 81, the one that was shown in that first trial. And I'm going to put this in grayscale because that's how it was shown. So the very first trial of this experiment of the natural scene stimulus was this picture of a shadow against a fence. And let's see, the second trial, you can also do this programmatically rather than typing in numbers, but the second trial was this cute picture of like puppies or wolves or whatever that is. So you can see that we have a lot of different natural images. You can, if you just want to see what the images are, actually the website is really useful for just flipping through them real fast, but you can access them all programmatically here and incorporate that into your code or into your analysis. And so I'm just we can take this information. So we want to look at the plot I made above of all the activity of those 50 cells. And now I'm just going to highlight whenever that first scene was shown, that we used in the first trial, the one of the shadow on the fence. And so I didn't define something. Oh, because I had given it a scene number. I kind of skipped a step above, but so yeah, so these are all of the times when we showed that first image of the fence with the shadow on it that you can see. So this falls into the 3 different epochs when we showed natural scenes. The epochs aren't the same. They're not even the same across the different experiments, which was maybe a mistake we made. But you can see all of the times when this is shown, and if you want, you can go in more closely and pull out a single cell and take the start and end of each of these trials and look at the activity of the cell during each of those trials and start doing some statistics and analysis. All right, in the last 2 minutes, I want to just show you a couple of key things. One is the metadata that comes along with in the experiment. So get metadata. This is a dictionary that gets returned that tells you a lot of information. Some of it you already know because you used it to identify the experiment. Other pieces, the only weird place you find it is in this metadata dictionary. So the sex of the mouse, the targeted structure, we already knew that. These ID numbers, we'd already figured those out. We also tell you here the time and date at which the experiment was collected, and we've had people do some analysis looking to see if there might be circadian effects on some of the responses that could use that. The age of the animal. There's also a device name. This is which exact microscope we collected the data on. We have 5 different microscopes that were used, and we've done some analyses comparing some of the activity across microscopes, making sure that we're not seeing differences based off of the different machines. So there's some nitty-gritty information in there that some of it might not be particularly relevant, but is good to be able to access. All right, I'm gonna just jump ahead so that I want to show one little thing before I turn it over to Josh. And this is how you can access the calcium movies. And again, you can only do this when you're in AWS, either in our portal or if you're accessing it through AWS through your own AWS account. We have the raw calcium movies stored in AWS. And so this code here, you're pointing it to a particular directory where we have these OFIS movies. And you can get the raw data path for a particular session. We've already identified our session ID. And this will create the path. And then you can access this data. And let's just plot it here. It's supposed to be animated, but maybe something's maybe not fully installed. But you can see this is the calcium movie that we just pulled up. And you can, if you're somebody who's interested in kind of how we do the cell segmentation, this is an area of active research in the field. We've got, 1,400, 1,500 hours of imaging data that is really rich for kind of working on those things. And there've been a lot of people that have used the data in this way. And I just want to make sure everybody sees how they can do that. If AWS isn't your thing, the other thing you can do is send us a hard drive and we can put movies on it and send it back to you. But really, you should get up to speed and use AWS for this because it's just a lot easier. Okay, at this, I'm going to wrap up and turn it over to Josh so he can tell you about accessing the Neuropixels data. So yeah, and if you have questions, throw them in the Q&A and I will go in and start answering any of the questions that have come up.
Awesome. Thanks a lot, Saskia. I'm going to share my screen. Okay, so this is the Neuropixels portion of the tutorial. There are some differences in the way that data is accessed or the Neuropixels data set. So I'll kind of walk through a similar set of steps that Saskia just did, but the instructions will be tailored to this particular data set. Some are just different conventions that we chose, but others are just due to the differences between imaging and electrophysiology data. So if you saw my talk this morning, you got a kind of a high-level overview of what's in this Neuropixels data set. So in short, we've recorded spiking activity from 6 different cortical visual areas, as well as a number of subcortical areas using Neuropixels probes. And we packaged all this data into NWV files and made it available through the Allen SDK. So within the this dev.dinebrain.org, you should, in the Open for Neuroscience 2021 folder, you should see 2 different Jupyter notebooks for Neuropixels. So one is the one that I am looking at right now, which has a number of blank cells, but there's also the one labeled answer key, which has all of the information already filled in. So if you're following along with the tutorial and you want to run the cells in tandem with me, it may be a little bit easier to use the answer key, which already has the information filled in. But you can also feel free to use the blank one and type in the commands as I am running through them. So this tutorial has 5 different parts. I'll start out with how to access the data, talk about the different information that's available for kind of browsing through the data and deciding what you want to analyze. Then we'll talk about how to access the data for a specific session and to read data out of NWV file. And then I'm not sure if we'll get to this today, but there's a section that shows you how to use subfunctions for aligning data to stimulus presentations. A lot of the analysis that we do is kind of the first step is making sure that the data is aligned to the start of a particular stimulus. And so we can look and see how activity changes from trial to trial, and kind of look at what is consistent across trials. And then there will be there's a short section on receptive field mapping, kind of how we align the spikes to our receptive field mapping stimulus and how we pull out receptive fields for individual cells. And then the last part goes into how it's like a short exercise on how to decode natural images using neural activity. Yeah, that's may be a good jumping off point for your own future analysis. So we'll get as far as we can in the allotted time, but the good thing about this tutorial is that the notebooks will remain active after the tutorial finishes, and so you can keep running through the different steps in the notebook. Just remember to shut down your session as soon as you're done, and so it doesn't cost us extra. So yeah, for data access, so all of the data is available through the Allen SDK. It's also hosted in some other locations, such as the Dandy repository, but really the easiest way to get up and running is to install the Allen SDK in your Python environment, or to use this AWS environment that we set up. And then there are commands that make it really simple to browse through the data and get access to NWB files, and you don't have to worry about kind of where the files are stored and like manage the downloads yourself. So yeah, everything, the interface for all the data is this EC-EFIS project cache object. And we use this term EC-EFIS, which stands for extracellular electrophysiology, because it's potentially more general than just Neuropixels data, but in this case, we've collected all the data with Neuropixels probes, so we kind of use those two terms interchangeably. The first step is to do the necessary imports, so these are already filled in, and then you have to decide where you want the data to be stored. So if you're running this on AWS, there is this path that's already predefined where all the data lives. I'm actually running this locally, so I have a path that points to a location on my local computer. One thing to be aware of here is that the files are quite large, so the NWV files are around 2 gigabytes just for the spiking data, and then if you want to download the LFP data, that's like sometimes as much as 7 gigabytes per session. So you want to make sure that if you're going to be accessing a large fraction of this data set, that you have ample space available on your local machine. But if you're running this on AWS, you don't have to worry about it. All that data is already on the cloud. So first step is to create this cache object that points to our manifest path. And then this will provide access to a number of files. And so these, similar to the 2P data set, there are these functions like get channels, get units, get probes that give you these give you access to kind of the metadata about all of the sessions and probes that we recorded from. So if we just want to see what sessions we have available, we can say cache get session table, and then so this gets loaded directly as a pandas data frame, and this is quite convenient because there's a lot of nice functions in pandas for filtering the data frame and kind of honing in on the data that you're interested in. And so yeah, if you're not familiar with the pandas library, I highly recommend doing some tutorials and learning more about that. We use pandas a lot in the Neuropixels portion of the Allen SDK, and you also saw some examples from Saskia. It's very useful library, so I highly recommend learning that. So this is just the first 5 entries in this table. We can see the information that's available, such as the specimen ID. This is the ID of the mouse that we recorded from, which type of session it was. So I mentioned that there were 2 different stimulus sets that were shown. One is called Brain Observatory 1.1, and the other is called functional connectivity. So you'll see about half the sessions with one of those 2 session types. You can see the age of the mouse at the time of the experiment, its genotype. So in our case, we're using transgenic mice to do optotagging. So we have some mice that express channel neuropsin in specific interneuron subpopulations, and then at the end of the experiment, we shine blue light on the cortex, and we can activate those cells and see which ones express the gene of interest. But we also record all the activity of the unlabeled cells. So this is kind of like an extra layer of cell type information that we get on top of the cells that we already record. You can get the number of units that were recorded for that session, as well as the number of probes and the structures that were recorded. So if there's a specific area that you're interested in, you can use this table to hone in on the experiments that have recordings from that area. But let's move on to the so for filtering these data frames, there's a kind of a standard syntax for filtering pandas data frames. You basically select the columns that you're interested in, and then say the values within those columns that you want, and then just use the ampersand to get the intersection of all these things. So this would give us a smaller table that contains only male mice with that express channel rhodopsin in somatostatin neurons, and use the Brain Observatory 1.1 stimulus, and include recordings from VisL, which is another name for LM, or lateral medial visual area. So this ended up with just 3 sessions, but there's still a ton of data in these 3 sessions. Yeah, that's just one example of how you might want to filter this data frame. So we also have a table that includes information about all the probes that were used. So each session includes data from up to 6 probes at once. So you can use this. Oops, get probes. So yeah, this is a table of all the probes that were used across all experiments. Some may actually be the same physical probe that's used in multiple experiments, but within each experiment, the individual probe gets a name, probe A, B, C, D, E, or F. And you can see the total number of units that were recorded by each probe, and which probes were in which areas. This is somewhat useful to pay attention to because typically the same probe targets the same area across each day. Kind of the probes are in a set configuration. So if you want to look at only the data from primary visual cortex, that's almost always from probe C. And so you could filter all the data that was recorded by probe C, and that will almost always include some cells from primary visual cortex. So I think that information is in the cheat sheet that's also available in the GitHub repository. Okay, next we can get information about the channels that were recorded. So yeah, this is within each probe. There are up to 384 channels. And so this is useful because we have, in almost all cases, we've mapped the channels to a particular location in the Allen Institute Common Coordinate Framework. And so you can say for each individual channel, not only what area is it in, but what precise location was it located in the brain. And so you can get this interesting spatial information and look at things like how receptive fields vary across different locations of the brain, see how things change based on depth in cortex or depth in hippocampus. So having information about the channel location is very useful. And then finally, we have our table of units. So this is the table that contains the metadata about all of the units that we recorded. So yeah, we start with the raw data sampled at 30 kilohertz. We extract units using kilosort, and then we apply some quality metrics to those units to kind of determine how well isolated they are, how much contamination they have, whether they're likely to be missing spikes. So I mean, one thing to be aware of is that there are varying levels of quality in these in every electrophysiology data set, and it's not so simple as dividing it into isolated single units and multi-unit activity. There's much more of a continuum here. So by default, we filter these units to only return ones that we would consider reasonable for most analyses. So we use these default filter values for ISI violations, amplitude cutoff, and presence ratio. There's a Jupyter notebook on the Allen SDK doc site that goes into much more detail about what these different what these quality metrics mean and why we've chosen these particular thresholds. But if you want to get access to every unit that is recorded, you can also do that by disabling these default filters. And so yeah, this will give us over 99,000 units. And so yeah, there's some analyses where you really just want to have access to all of the information about activity in a particular area. Like if you're doing a decoding analysis, you may not care if all of the cells are well isolated or not. But if you're doing an analysis of, say, bursting activity, and you really need to know exactly when every spike for a particular unit happened, and you want to be sure that you're not missing any spikes, then you want to be more conservative in your quality metric thresholds, and maybe even use ones that are a bit more stringent than the default values. Okay, so to wrap up the data access portion, I'm just going to make a pretty plot using the information in the units table for the spatial information for every unit that we've recorded. And so here, yeah, you can see we are sampling densely in cortex, hippocampus, and thalamus. The colors indicate the area that each of the cells is mapped to. So yeah, this gives you a sense of the scale of the data set, and hopefully gets you excited to dive into it a bit further. Okay, so those are kind of the tables that you can get that give you information about all experiments, but a lot of the analysis will require accessing data about spike times or local field potential, and those come from NWV files that are specific to one session. So if we go back to our the data frame that we pulled out of the sessions data frame, we can choose one of these sessions. So the index of that data frame is the session ID, and so in this case we're just pulling out the third index because the numbering starts at 0 in Python, and then loading this session using another function within this cache object called get session data. So that takes the session ID as input, and you can get that session ID by filtering the sessions data frame. Okay, so within each of these sessions, there is a units table. So this sometimes takes a second to read in all the information, but basically this will give you the metadata about all of the units that were recorded in this one session. And then that table will have as its index unique ID for every unit. And then you can pull out the spike times, et cetera, for each of those units. And the unit IDs within the NWB files are the same as the unit IDs that we originally pulled out in this very long table of units. So if you find a unit in this larger table that looks like it could be interesting, you can then figure out what session it's in and then load the data for that session and then access the information for those units. Okay, this is taking a second. Okay, so here we go. So yeah, this has similar information that was in the original units table. Some, like for each unit, there's some information about its waveform. So if you're looking for example, fast spiking cells in cortex, you can use some of those waveform metrics to pull those out. It also has information about the structure that it's in. This is anterior pretectal nucleus in the midbrain, and then what probe it's on, etc. So we can filter this table in a similar way to the way we filter the sessions table. So here we're taking all the units in primary visual cortex and then setting a bit of a more stringent cutoff on ISI violations. So these would be considered units in primary visual cortex that have lower contamination. So if this results in 35 units from this experiment. And then we can get the unit ID by looking at the index of this table. So this column on the left is the unit ID. We'll just take the first unit from this table, and then we call session.spike times to load in those spike times. And so one thing you'll notice here is that it gives this warning that said there are some invalid times in this session. This is not true for most sessions, but there are a few sessions that do have invalid times. Basically, it meant that there's something that went wrong with the data acquisition, and we want you to be aware of that. So basically, like, because we have 6 probes requiring data at once, sometimes there's a problem with one probe, and just because that there's a short period of time when that probe's data is invalid doesn't mean that we want to throw out the entire session, because there's tons of data there. But this is something that's important to be aware of. But in order to figure out what went wrong, you can just type session.invalid times, and in a second it should show what the issue was. So in this case, there were about 20 seconds where probe E went offline. In our case, since we're analyzing B1, it's probe C, so we don't have to worry about this. But in general, this is not something you have to worry about, but we do want you to be aware of it in case you see like a section with no data, with no spikes. That doesn't necessarily mean that the cortex went silent for that period. It could mean that there's just data missing for that interval. Okay, so another thing you may be interested in is the waveforms for particular units. So we can load those just by using session.mean waveforms. And then the Allen SDK also includes a function called plot mean waveforms that will plot those for you. It's very self-explanatory. So in a second, this should pop up. And so yeah, the waveforms are actually saved for all of the channels on the Neuropixels probe. So you can see kind of this 2-dimensional waveform with lots of information about how the spike is propagating through the brain. But in this case, we're just going to pick out the peaks channel, so you can see the overall shape of the waveform. Okay, well, there we go. Okay, so yeah, here you can see the unit IDs as different indicated with different colors. You can see a combination of these fast spiking cells with a narrow peak to trough interval, and others with a more regular spiking waveform shape. Okay, so I will talk about how to access the stimulus presentation times, and then I think we'll probably move to Q&A. So similar to the 2P data, you can pull out the names of the stimuli. They do have specific names that you need to remember in order to filter the stimulus presentation table. That will come up in a second, but the using session.get stimulus table, you can get the stimulus table for a specific stimulus type, such as drifting gratings, and that will give you the information about all the presentations of that particular stimulus. Let's do one more section, which is the behavioral data. So you can also get the running speed for these sessions. It's just session.running speed, and that returns a table that has information about the start time at which the running speed interval was recorded, and then the velocity of the mouse at that time. And that will allow you to visualize and analyze the running speed. And there are definitely cells that are modulated by running speed. The responses in cortex can be quite different depending on whether the mouse is running or stationary. So that is something that is interesting to analyze. And then you can get information about the mouse's gaze location, both the size of the pupil as well as the location that it was pointing. I'm not sure why this is taking so long. Usually this function is quite fast. In any case, I think we can move on to question and answer. It's about 17 minutes left in the tutorial.
Great. Well, to get us started with questions, we have one that came in on YouTube, and you'll see there is a response to this question that we have posted in the Zoom Q&A, but we'd like to review that answer. So the question is, does this mean that I can't access the data without an AWS account? Also, are there sample data sets, 2P or EFIS, that one can test their own pipeline on? Saskia?
So you can access the data without AWS. You just pip install Allen SDK. You can use the SDK directly on your machine. It will download the data to your machine, and you have access to all of the NWB files and all of that. The one piece of data, at least for the calcium imaging, the one piece of data that you cannot access that way is the movies, the calcium movies. That's the only chunk of data that we can't make available that way, just because those files are just way too big. And so if you want to run your own processing pipeline on the calcium movies, you either need to do it on AWS, or like I said before, you can send us a hard drive, tell us which movies you want. We can put the movies on the hard drive and send it back to you. We have done that for people. We definitely prefer to do things via AWS, but we will do that if that's something that you need. So just reach out to us and we can make that happen. But everything else for calcium imaging, Josh, I don't know about raw traces for the Neuropixels.
It's on AWS too.
Yeah. So it's a parallel thing there, but everything else is just NWB files, and you can use that locally.
So it looks like there are two questions in the Q&A. Richard, first question is that the channels on Neuropixels probes are very closely spaced. It's about 20 micron vertical spacing between the individual channels. And so the question is, how do you then assign a position on the probe to an individual unit? So we basically just do that by looking for which channel has the highest amplitude response, and we assume that that's where the cell body is likely to be located. There is, yeah, I mean, we've done some analysis on the spatial extent of these waveforms. They kind of spread over 100 microns or so of the probe. So there is some ambiguity as to exactly where the cell is located, but in order to assign a specific location, we just pick the channel that has the largest amplitude waveform and call that the peak channel. And then that's where we say that the unit is located. And then another question from Richard is what one might really like is a GUI that would allow the user to select data and then display, for example, a raster plot. Does anything like this exist? Yes, actually. So the NWB development team, I think led by Ben Dichter and Matt McCormick, have created a library called NWB Jupyter widgets. And if you install that and point it to an NWB file, it allows you to browse through the data in that NWB file and create kind of summary plots of what's in there. And they've actually created a lot of nice widgets specific for Neuropixels data, which allow you to do exactly that, like pick a segment of time and a certain set of cells and then plot a raster plot. So I definitely encourage you to take a look at that if you're interested in kind of quick ways to dive into the data and browse through it. There's another question about whether it's possible to access simultaneous EFIS and calcium time series from a single cell. And so, yes, we have, it's a much smaller data set because these experiments are a lot harder. But if you go to brainmap.org and browse to, there's a page for all of our calcium imaging and Neuropixels data. And then there's a link to where you can download the files that have the simultaneous recorded EFIS and OFIS data. So
we have another question that just came in from YouTube. What are the strategies you're using to decode EFIS data into a corresponding correlated natural image stimuli?
So yeah, I mean, there's a lot of different strategies you can use. I think we tend to start with kind of simplest possible way of doing it. Even sometimes a linear decoder is enough to be able to decode stimulus identity. Find that random forest decoders also work quite well, but then yeah, you can definitely get into deep learning strategies and kind of throw whatever you want at it to see if you can do better. But yeah, kind of our go-to one is a random forest decoder, but you can use almost anything.
So to return to another earlier question, this one was directly to Saskia, but I think it would be good to confirm this parameter for Neuropixels as well. How does one figure out the degree of visual angle, subtended by any given stimulus? And Saskia, you already answered this one, but maybe we can review that and cover it for Josh as well.
Yeah, so there's a lot of information on the details of kind of the monitors, how they're calibrated, how they're positioned, that you'll find in our white paper. Some of the information is also in the methods section of the papers that we publish using these data sets, but the white paper is really much more extensive in this. And it talks about, so the simple answer is that the monitor subtends 120 degrees horizontally, 95 degrees vertically. The stimuli that we show, for instance, the gratings that we show are all full field gratings. And so they fill that monitor space. We do warp the stimuli. We do a spherical warping. And so like I said, there's details about how that's done and what that looks like in the white paper that explains all of that as well. So I think in my talk this morning, I think I pointed to where this documentation is. But again, if you go to the brain map, if you go to the observatory.brainmap.org, there's a link there for documentation. We have like 4 or 5 different white papers. One's on the stimulus set and analysis that has a lot of this information about the stimulus protocol.
Yeah, and similar parameters apply to the EFIS data set. We replicated pretty much exactly the monitor position and all of the different parameters of the rig related to the visual stimulus. So Pretty much anything that's true about the 2P data set also applies to the Neuropixels data set. And in cases where stimuli were shown for a smaller region of visual space, so like the Gabor stimulus that we use for receptive field mapping, so all of those, the values for like the stimulus size are already translated into degrees of visual angle. So we have a 20-degree grading stimulus, which is 20 degrees from the mouse's perspective anywhere on the screen, because we do this spherical warping to account for distortion at the edges of the screen.
And this is a question that we commonly get about the stimuli, is how would someone go about accessing those?
So, I mean, if you are interested in the natural movies and the natural scenes, so those templates are available via the Allen SDK. I think Saskia showed an example of how to download the original images and movies that were shown. If it's things like the drifting gratings, those were displayed using the PsychoPy library, and in that case, if you want to show them yourself, you can use PsychoPy to generate the stimuli and show them with the same parameters that we used. But in those cases, you kind of just have to trust that we got the stimulus right. But all the parameters that would be necessary to replicate those stimuli in PsychoPy are available in the white papers.
Great. We do have time for a few more questions. So while you think about what you want to ask, just a quick PSA about AWS. So for those of you who were following along using AWS, please do clean that up. When you're not actively using the notebook, please stop it. So you can go back to that main page and hit stop the notebook, and then you can restart it. As Saskia mentioned, you can also save a local copy of that notebook so you can access it later. And please make sure to, after you've saved everything that you want to keep, delete those stopped notebooks. So this will prevent using those AWS resources. Saskia, anything to add to that?
No, I think, yeah, that covers it. If you're not going to be using the notebook for a while, I would suggest stopping it. And when you're done, delete it, just because the instances cost money. And so this will only be available for a little while. So, if in the rest of the day you want to keep looking at the notebooks and trying some new things, by all means do it. Feel free to download the notebooks. I pointed you to the GitHub repo where you can just access the notebooks directly. And there's solution sets for both the Neuropixels and the 2P that kind of have everything already filled in. So all of that's available. And I think Josh and I both are feel free to hit us up with questions. The community forum is a great place to put questions and see questions that other people have already asked. And we reply there. You can also email us directly, and we're always happy to help people get up and running using the data. So feel free to reach out.
And we will post also a link to the community forum and the links to the notebooks in the video description of this on YouTube. All right, let's see. I don't see any additional questions. Do you have any maybe final FAQs that you'd like to preemptively answer?
None are coming to the top of my mind at the moment. I think a lot of things that people run into really just comes with familiarity with the SDK and how we've kind of structured the data set. I think, with the 2-photon data, I think one of the places where people sometimes get, work with is when you're looking at a cell across different sessions. One of the things I didn't have time to explain today is we have this unique cell ID and how you can use that to either identify which experiment you want, but even within an experiment, kind of how you link that ID to a particular trace. And you can use that ID to get the index in that array object. And that index will be different in session A as it will be for session B if you have the same cell in both sessions, right? And so you have to do that kind of within each session separately. And that's one of the challenges with the 2-photon data because it is split up into multiple sessions. And I can send some information about that. And there's some information on the cheat sheet about that as well. So.
Yeah. And if you'd like a deeper dive on both of these data sets and more than we can do in an hour and a half, applications are currently open for the summer workshop on the dynamic brain. Those are open this time of year every year. You can find more information by going to the Allen Institute website and clicking on events. More information and the application there. All right, well, it looks like we have answered all of the questions. Again, if there's any further questions, please post them in the community forum and we'll answer them after the fact. All right, well, thank you everyone for being here, and thanks to Saskia and Josh. And I hope we'll see you tomorrow at the last day of the Open for Neuroscience Symposium. And please check out some of the other tutorials in this series to learn more about other open resources from the Allen Institute. Thank you, everyone.
Thanks, everyone.